Dear students, welcome to 12th class Biobotany. So we were discussing about the chapter Asexual and Sexual Reproduction in Plants. So last class we studied about tiers of anther and what are the different layers of anther wall, epidermis, endothelium, middle layer, tapetum and their function. Today we are going to study about pollen grains. Okay, so how these pollen grains are developed in this anther, you already know. Inside the anther, the sporogenous cells, they undergo mitotic division and they form microspore mother cell. Microspore mother cell. Okay, they forms mitotic mother cell by mitotic division, and this microspore mother cell divide by meiotically and form microspores, haploid microspores. Microspore mother cell is diploid, but by meiotic division they produce microspores which are haploid. From these microspores, what develops? Pollen grains are develops. Okay, from microspores, pollen grains are developed. So you already know these steps. So let's check what is the structure of pollen grain. Pollen grain which has dense cytoplasm and pollen ball. So this is the structure of pollen grain. They have dense cytoplasm and which is surrounded by pollen ball, and this pollen ball which is differentiated into two layers. Okay, what are two layers? The outer layer is called exine and inner layer is called indine. So this is a pollen ball, and pollen ball has two layers. Outer layer is called exine, inner layer is called indine. Okay, so let's discuss what is indine. Indine we already know, which is inner layer of pollen grain, indine. So you can see here this is indine. This indine which is very thin, and uniform in structure. Okay, which is thin and uniform, and it is made up of hemicellulose pectin. It is made up of hemicellulose pectin, cellulose and callose together. So pectin, this indine inner layer is made up of hemicellulose, pectin, cellulose, callose together, and this indine which is uniform in shape, in structure, and which is made up, which is thin layer. Okay. Then what about exine? Exine is outer layer. Exine which is thick, outer layer which is thick. Okay, and exine is made up of cellulose, sporopollen and pollen kit. So exine, the outer layer which is made up of sporopollen, cellulose and pollen kit, and which is thick. Okay, exine, outer layer which is thick. So you already know what are the difference between indine and exine. Indine is thin layer and which is uniform. Exine is thick and which is not uniform. In structure. Okay. So indine is uniform and indine is thin and made up of hemicellulose, cellulose, callose and pectin. But exine, the outer layer which is thick and not in uniform, and which is made up of cellulose, sporopollen and pollen kit. But in case of exine, in some areas you can see they are thick. This area they are thick, but this area, some areas they are thick. In some areas exine is thin. In some area exine is thin. Okay. In those areas they form a small round structure, round pores. Okay, those pores are called germ pores. In these thin areas on the exine, germ pores are present. What is germ pore? on the small on the thin area of the exine, small round opening is formed, that is called germ pore. Okay, if this thin area, when it is elongated, it is called furrow. So these germ pores, that openings, that pores present on the exine, which helps pollen germination. So germ pore which helps in pollen germination. So these are the pores present on the exine, which is present on the thin area of the exine. Okay. Exine which is not in uniform and they have germ pores. Okay. These germ pores which helps in formation of pollen, which helps in pollen germination. On this germ pore, sporopollenin is absent. So here in this germ pore, on this germ pores, sporopollenin is not present. Okay. Then pollen exine, which may be sometimes smooth or which may be sculptured. So pollen exine surface which show different sculpture patterns. They may be rod or wave or groove, or maybe vati. So different or various sculpturing patterns are present on this exine, pollen exine. So based on different or various sculpture patterns, uh, we can identify the plants and also we can classify the plants based on different sculpturing patterns present on the exine. So pollen exine may be smooth or may be sculptured. Okay. And pollen size also different. Pollen size may be globoid shape, or maybe like ellipsoid, or maybe angular or lobed. Okay, from different one species to another species, the pollen grains size is different, shape is different, and size also different. That size, pollen size may be 10 micrometer to 200 micrometer. Okay, for example, in Cucurbitaceae, like pumpkins, their family, the pollen grain sizes are around 200 micrometer. Okay, so pollen, they have different shape and they have different size, one species to another species. So this point you have to keep in mind. So what is sporopollenin? The exine, which is made up of three components. What are those three components? Cellulose, sporopollenin and pollen kit. So sporopollenin present on the exine. So what is actually sporopollenin? Which is derived from carotenoids. Okay, and these components for sporopollenin, which are contributed by pollen cytoplasm and also tapetum. So we discussed already about tapetum and their function. At that time I told you the tapetum which contributes for the formation of sporopollenin and pollen kit. Okay, so here this sporopollenin, it is formed by with the help of pollen cytoplasm and tapetum. Then what are the features of sporopollenin? The sporopollenin which is resistant to physical and biological decomposition. Okay, so it prevents the pollen grains from decay and decomposition, and this sporopollenin which can withstand higher temperature and action of strong acid. So if it is present on the pollen grain surface, that pollen grain can withstand higher temperature and also strong acid reactions. And it also preserve the pollen grains in fossil deposits and also at the time of pollination from anther to stigma. Okay, so these are the main functions of sporopollenin. They are derived from carotenoids and they are contributed by pollen cytoplasm and tapetum, and they can withstand higher temperature and strong acid, and which uh, which are resistant to physical and biological decomposition, and which preserve the pollen grains in fossil deposits and also the time of pollination from anther to stigma. Okay, so this is the component present on the exine, which is this sporopollenin. Then next component present on the exine, which is pollen kit. Pollen kit. So exine is made up of cellulose, sporopollenin and pollen kit. So what is actually pollen kit? Pollen kit, which is something like a oily layer or a thick viscous layer coating, a thick viscous coating on the pollen surface. Okay, and which is chemically these pollen kit are made up of carotenoids or flavonoids, and these components they are contributed by tapetum. So at the time of tapetum function, the second function is tapetum contribute for the formation of pollen kit. Okay, then what are the functions of pollen kit? Pollen kit which attract insects, especially for the pollination, and also which protect the pollen from the UV radiation. So these are the main functions of pollen kit. So let's check pollen kit. It is contributed by tapetum, we already said, and this is something like a colored yellow or orange. They have yellow or orange color, and 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 they are derived from carotenoids or flavonoids, and it is something like a oily layer or a thick viscous coating on the pollen surface, and which attract insects and which protect this pollen from UV radiations. Okay, so these are about sporopollenin and pollen kit. So when these pollen grains are liberated from anther to stigma, if they are reach or land on the right stigma, these pollen grains they try to germinate. Or if they are not landed, if they are landed on the wrong stigma, they are rejected. Okay. So pollen grains they land on the right stigma and they swell and they take some moisture and they swell up and they forms pollen tube. What is pollen tube? Actually, the inner layer which form pollen tube. So the contents present in the pollen grain, that is generative cell, and that generative cell it comes through the pollen tube. Okay. Then this generative cell which divides into two male gametes, or two sperms. So what happens? The pollen which land on the stigma, the pollen which forms pollen tube, into this pollen tube what comes? Our generative cell come, and this generative cell divides into two male gametes. Okay, before reaching to ovary. So this is about male gametophyte. So finally what happens? Two male gametes are developed.